Today we're going to be going over enzymes. So to start the class, I would welcome all the students in, as they come in. If there was anything to take care of from yesterday, I would take care of that. And then I would start the lesson with this video about enzymes and how they are compared to a snowman. So I would pull this video up and play it.
Making a snowman is always a big ball of fun. 3 snowballs full of fun to be exact. And each snowball starts out small, getting bigger as you roll it in white fluffiness. Let's imagine that you go outside in a fresh snowfall to build one of these guys, but you don't have the best suited yard for rolling snowballs. In fact, your backyard is one giant hill. This hill is fantastic for sledding, but not so much for making snowmen. Anyways, in order to make a giant snowball for the snowman's bottom, you'll need to actually push this snowball up over the hill and then down the other side. You do it because it's fun, but make no mistake, it takes a lot of energy to get started up this hill, and this is hard work. Right now, you share a quintessential problem with some chemical reactions within a cell. These chemical reactions might be a lot of fun when you're in a cell, but it takes a lot of energy to get them started. This is called the activation energy, or the energy required for a reaction to start. You can think of activation energy as that energy you needed to muster up in order to push that snowball up and over the hill in your backyard in order to maximize the size of your snowball and get it to the other side. What your cells have to help overcome a problem of high activation energy are called enzymes. Enzymes are proteins that lower the activation energy of a reaction. In doing this, enzymes increase the rate of a reaction, helping it to occur faster. However, enzymes are not consumed in a reaction, they simply help it to occur. They do not add snow to your snowball, they just make it easier to make a snowball. This makes enzymes reaction catalysts. The presence of enzymes is analogous to decreasing the size of your hill in your backyard. Enzymes make things easier for your cell and help chemical reactions occur. There are hundreds of different kinds of enzymes in your cells, which all participate in different types of reactions. Enzymes can break molecules apart, build or add molecules, and even rearrange them. In lowering the activation energy of a reaction, enzymes decrease the barrier to starting a reaction. It's important to note, however, that the change in energy remains the same between the start and end of a chemical reaction. For example, if a reaction released 200 kilojoules of energy without an enzyme, the same reaction would still release 200 kilojoules of energy with some enzymatic aid. The difference would only be a lower activation energy and a faster rate of reaction. In other words, if enzymes were helping you build a snowman, you'd be done quicker, but you'd have just as much fun.
So after this video, I would discuss it with the students, and then I would go into the PowerPoint presentation over enzymes. Sorry, you got a double shot of me. Presents down here. Okay, so today we'll be going over enzymes. And first we'll talk about catalysts. Like mentioned in the video, they kind of speed up a chemical reaction, but it's not consumed in the reaction. So it's kind of like an assistant. So they provide alternate pathways for reactions to occur by lowering activation energy, gives more molecules with required activation energy. So there's more collisions a second and there's a faster reaction. So inhibitors, some definitions. Inhibitors reduce the rate of reaction. The transition state is the highest point of the activation energy required through bond breaking. The activated complex is the short-term formation of bonds after transition state of bond breaking. And the activation energy is the amount of energy required for products to form. And then I would show them on the graph where everything was located and give them time to copy this down, ask if there were any questions over what a catalyst was or what any of the definitions meant, and then explain them if there were any questions. And then I would talk about that enzymes are sometimes considered an exothermic reaction. So they release heat energy. So an example of this would be cellular respiration, which breaks its own glucose to make cellular energy. And I would explain the graph and what everything meant and give them time to ask questions and to copy everything down. So you have the activation energy with no catalyst, it's a lot higher than the activation energy with the catalyst, and then heat is given off because of the catalyst. And then we also have endothermic reactions that absorb heat energy. So an example of this would be photosynthesis, which absorbs sunlight to create glucose for plant energy. So the activation energy is again a lot higher, and then once there's a catalyst, the activation energy needed decreases, and then heat is taken in rather than put off. And then enzymes are biological catalysts. So a substrate plus an enzyme will produce an enzyme, will combine with the substrate, and then they'll break apart with the product. So there's acid-base catalysts, covalent catalysts, electrostatic catalysts, proximity orientation, which are molecules colliding when they're nearby, and then large molecules containing carbon, hydrogen, nitrogen, or oxygen. There's substrate specific, and there's an active site where the substrate binds, forming intermediate that lowers the activation energy. And there are general properties of catalysts, so it speeds up the rate, but it is not consumed. An enzyme provides a template where the substrates are brought together in proper position to react with each other. A substrate is a molecule on which an enzyme acts. So on the picture below, the green piece is the substrate entering the active site of the enzyme. The enzyme and the substrate complex combine. So the enzyme changes shape as the substrate binds, and then the enzyme products complex, and then the products leave the active site of the enzyme. And then there's a lock and key. These are different types of enzymes. There's lock and key where the substrate fits precisely into active site of enzyme. There's an induced fit. Enzyme and substrate configurations are modified by substrate binding. Conformation of substrate is altered so it resembles that of transition state. Stress produced by distortion of substrate can facilitate conversion to transition state by weakening critical bonds, and then the transition state is stabilized by tight bonding to enzyme, so decrease in activation energy. And this is a picture of the lock and key model, so it fits like a key in a lock. And then the induced fit, the molecule changes to fit the enzyme, so it changes completely. And then there's non-biological catalysts, so some small molecules such as metals, they have large number of electrons which don't mind how close to the central atom they are. This allows metals to use these electrons to help out in reactions before claiming back once the reaction is over. Formation of weak intermolecular bonds followed by chemical bond formation, minimal specificity for substrate, and easy separation from products. And then these would be the review questions that we would answer and then go over. So how does an enzyme affect the activation energy of a chemical reaction? I would give the students a few minutes to answer this question, even work with their elbow neighbors if they would like, and we would talk about how the enzyme lowers the activation energy of a reaction, making it more energy efficient. And then how does an enzyme recognize its substrate? We would talk, again, I would give them time to answer this question and work with their elbow neighbors if they would like. And we would talk about how there are specific shapes that would need to fit, or chemical reactions that would need to be done to make it fit. And then name 2 ways that a molecule may inhibit an enzyme. So it could, another enzyme could fit the space that it doesn't need to, or it would have something in its way of being connected. And then after the slideshow, the students would do a lab. So I would have these in printed off versions, of course. But for the function of this, we'll just go through this. So they would be doing a mini lab, observing enzymes at work. So I would go over the introduction with them, the objective, safety, and materials and procedures before letting them get in their groups. So the introduction, an enzyme is a biological catalyst. A catalyst is a molecule that speeds a chemical reaction without being destroyed or used up in the reaction. The enzyme you'll be working with today is catalase. Catalase is an enzyme found in most living organisms. Its function is to break down hydrogen peroxide into the safer compounds of oxygen and water. When catalase breaks down hydrogen peroxide, it forms bubbles. You've probably observed this when you use hydrogen peroxide to clean a cut. Human cells contain catalase. So the objective for this lab is to observe the effects of catalase on hydrogen peroxide and to observe enzyme action in living cells. And then I would remind them of the safety procedures. Do not eat or drink anything in lab. Please report all spills to me. And goggles must be worn at all times until everyone has cleaned their work area. So materials would already be set out, and there'd be weigh boats, medicine cups labeled 1, 2, 3, a milliliter plastic pipette, peroxide, catalase, a cooked potato slice, an uncooked potato slice, vinegar, and water. And then I would go through the procedures with them, which part 1 states that you'd obtain a fresh slice of potato and a cooked slice of potato from me. And place the cooked potato and the raw potato in the weigh boat. Place a dropper full of hydrogen peroxide, so 1 milliliter, onto each potato and observe what happens. Record the observation below. And then for part 2, add 3 milliliters of hydrogen peroxide and 3 milliliters of water to medicine cup 1. Add 3 milliliters of hydrogen peroxide to your medicine cup and 1 milliliter of water to medicine cup number 2. Add 3 milliliters of hydrogen peroxide and 1 milliliter of vinegar, which is an acid, to cup number 3. Add 2 milliliters of catalase solution to cups 2 and 3. Cup 1 does not get any catalase. And record your observations in the table below. And then when they were finished with the lab, I would instruct them to clean up and how to clean up, and then they would answer these analysis questions. So what is the purpose of catalase in living systems? Why did the bubbles form on the raw potatoes but not on the cooked potato? What was the purpose of the water in part number 2? And explain the results of part 2 and why did the reaction not occur in cup 1 or 3. And while they were doing the lab, I would be walking around making sure there weren't any questions and that all safety procedures would be being followed. And then once they were done with this, they would have a quiz to do, which is over enzymes and is 5 questions long. The first question is, enzymes are proteins that help increase the rate of chemical reactions inside cells. These proteins are composed of many simpler molecules called amino acids. Which of the following suggests that the shape of an enzyme determines the enzyme function? And then number 2, sucrose is broken down in glucose and fructose in a metabolic reaction. Which statement best describes the role of an enzyme during this reaction? And then number 3, a scientist observes that when the pH of an environment surrounding an enzyme is changed, the rate catalyzing the reaction significantly decreases. Which statement best describes how a change in pH can affect an enzyme? Question 4, which of the following molecules is an enzyme? And then number 5, in the potato enzyme lab, the enzyme was the blank and the substrate was the blank. And then for any GT extension, or if they finish early, there would be a card sort activity that they could do on their devices or to do at home if they would like. And then That would be the lesson that they would be given that day. At the end of class, I would go over the quiz to make sure everyone understood what we were expected to know for enzymes and make sure there weren't any questions.